Hi, my name is Kaylee Guzman, and today I'll be talking about the melanocortin 1 receptor, also known as the MC1R, which is what we are studying in BIOC 108. The location of MC1R is found in the plasma membrane of the melanocytes, which are the cells that are responsible for skin, hair, and eye pigmentation. The connection to the disease that we are studying is a mutation in the MC1R, which can lead to red hair, fair skin, and increased risk of skin cancer, which is due to reduced production of eumelanin, which is a dark pigment, and an increase in phelomenin, which is a light pigment. As you can see, the mutation binds to the receptor and it changes it, so it creates it as a mutation that can lead to red hair, fair skin, and an increase of skin cancer. And it's detached over here. The overall shape of the MC1R is a G-coupled protein with a characteristic of a 7-trans membrane helix structure, and so this bands in the plasma membrane in a serpentine manner. So this is what I did here, making it like that. And in the extracellular region, it is involved in a ligand binding, while the intracellular part interacts with signaling proteins. So that's just demonstrating the structure and how the inside binds with the signaling powers, and the extracellular is involved with ligand binding. The materials I used for this specific structure was beads and string in order to show the helix structure, but also the different mutations and how that binds to it as well. For the structure of MC1R, there could be a few different structures, which is the N-terminus, the extracellular, which is important for ligand binding, the 7 helicase, which is what I'm demonstrating in my structure, spanning the membrane, forming the receptor's core structure, 3 extracellular loops, helping with ligand interactions, and 3 intracellular loops by interacting with the G proteins. And then the C-terminus, which plays a role in the receptor regulation, which would be this. So MC1R functions as a monomer, so it's a single unit, so it's a singular continuous chain rather than being or having multiple subunits. So in this structure, it's a primary structure, so it's a sequence of amino acids, which would be linear, but also secondary structure that is almost like a helicase shape, and a tertiary structure, which is 3D. So this is a tertiary structure, which is 3D, just because it's showing how the mutations bind in the different helix structures. It does not have a quaternary structure. So the mutations can affect the structures by disrupting its ability to bind to ligands or impairing cell surface expression, what I demonstrated earlier, where the different mutations will bind to it, and so this can lead to a structural instability or misfolding, which prevents it from properly functioning, and so that is how the mutation affects the function, where a mutation can reduce the MC1R's ability to activate signaling pathways, leading to less eumelanin, which is dark pigment production, that will lead to fair skin and red hair. It can produce more pheomelanin, which is light pigment production, which is an increase in UV sensitivity. There could be a higher risk of skin cancer risk due to less or natural UV protection. And then in many MC1R mutations, there is an occurrence in the transmembrane or intercellular regions. And so then that's what we showed earlier, or right here, where the mutation binds and prevents it from working properly. Some limitations is that the model clearly does not capture atomic level interactions, such as how the MC1R is binding to the signaling molecule, so that would be better, and maybe like a version of an online structure. However, this is just a simple model. Also, a static representation where MC1R is a dynamic protein, but this model shows it as a fixed form, so it doesn't show its movement, and the simplification of its membrane environment, so the real protein is in a fluid lipid bilayer, but the model doesn't accurately represent how it interacts with the membrane. Thank you so much for listening.